yeah, where we can have language models. Okay, so I'll come back to this. Yes, Sayak Mandal, I'll come back to this. If this thing repeatedly happens, we'll change the protein. No worry for that. And then we'll subsequently see what exactly happened. Okay, so what happened? I received one this thing feedback that the PS is very fast and so much of background information is needed to assimilate all the topics that I covered, and even my discussions many a times are uh, 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 um, with so much of shall I say info that some background information is needed. So what exactly we are doing is we are trying to inculcate among biomedical scientists. I'm talking about myself. My idea is to inculcate this application of computer technology, or let us say microprocessor technology, among biomedical scientists who may have, yeah, yes, yes, I have come across this. We'll get we'll be doing this later on. So this was the idea. Okay, I hope the other student who had some difficulty is there in the this session. So in order to understand all this, we should understand. Okay, we should understand that the overall goal of microprocessor technology is to predict. Okay, the final goal, the ultimate goal of all computer technology is not only to write a letter in MS Word or do some graphic design in MS PowerPoint or things. The ultimate goal of computer science is to predict. Okay, this today's session will be a bit longer. Do not worry for that. So I tell you about this thing. So long back, what happened? You know, when computer was not there, we were not as advanced as this. So people devised a strategy to predict very simple things. To predict what is 2 plus 2, what is 2 plus 3, very basic calculations. Okay, so in order to do that, what the strategy they applied is because they were designing a machine. So what they were designing was they were designing some levers. Okay, lever, you know, lever means one circle completes, then it is connected with one more circle, and therefore along with the first circle the second circle also moves. Okay, something like inside your watches, this kind of lever mechanisms are there. So fast computers are nothing but the lever mechanism, what you also get in your analog watches, not smart watches, analog watches. So the ultimate goal was that. Okay, later on it was been found that there are multiple applications, and eventually MS Word, Excel, and those things were there. Then video streaming services and multiple other things. The ultimate goal of computer science is to predict. So you can contradict. You can do a little this thing that why sir you are saying like this, because computer science has been useful for many things. I write my project in this, multiple other. I may not able to design a software, but I'm using that. But you know, this is not the ultimate goal. The ultimate goal was to predict. Okay. For example, these days the language models are there. I do not know. I think many of you are aware that there are some artificial intelligence tools which are basically advanced language processing. Okay. So how I tell you, there is a tool, as you all know, called Mid Journey. Okay. Now, in Mid Journey, what you can do is you can design any kind of landscape. But all your wildest dreams can come true in Mid Journey. Why? Because it can predict multiple levels of results only by a simple input, like imagine draw a flower in which one this thing a butterfly is sitting, and inside that butterfly there is a bug, inside that bug there is a bacteria, inside that bacteria there is a lysosome or whatever, then inside that there is a protein, inside the protein there is an amino acid. So if you prompt this thing in Midjourney, Midjourney will eventually give you a result. So what exactly is doing? It is only processing the language, and on the basis of the very simple instruction it is able to predict what is the what kind of image you want to get. Okay. So how all these things started? Okay, we'll just check it out. As I have said, machine learning is or data science or artificial intelligence. These are nothing but a tool to predict. Now, this prediction needs a multiple type of algorithm. Now, algorithms are nothing but instructions that you give to a computer. Okay. Now, computer has the ability to follow instructions. For example, if you press the keyboard digit A, then it will always give you the digit A on the screen. Okay, exactly like this, because the computer has been instructed to do so. Okay, so we'll see first of all that how exactly this predictions takes place. Okay, what is the how you can do the predictions. Okay, the base kind of prediction is done by means of a technique or a statistical tool which is called as regression analysis. Okay, so we have already spoken about it. To understand this further more, I will request our coordinator madam to click on the Excel file and share the Excel file and repeat the tasks so that our participants can one more time understand what exactly is going on. If it is already there and if it is already done, the same file you can open. I'll just once again repeat so that our participant who was not very confident can able to assimilate what exactly is going on. In that what I'll do is we will able to, do not worry, if we will definitely do some tools also, and the troubleshooting will also be done. Why exactly that this amino acid was not getting identified, we will check it out. So in that Excel file, what exactly we did is we have taken two training sets. Okay, two training sets, one in x-axis, another is in y-axis. So you may ask me why training sets are needed. Coordinator madam, you are there? Coordinator madam?
Hello. Yes, sir.
So what exactly is going on is we are having two training sets. Okay. So training sets are nothing but your experience. Okay. As I have said, no. I want that Excel file, madam. I'll come back to this later. I want you to minimize it
Yeah.
and share the Excel file. So what exactly happened is, first of all, we'll talk about data science. Okay. So data science is nothing but accumulation of or a cumulative of all sorts of statistics that is already there in human conscience. Okay, all sorts of statistics. It could be mean, it could be median, calculation of mode, calculation of averages. So all these things together, when implemented in a software, we'll call it data science. And more so if you are using a huge amount of data for the same calculation. Okay, so you can tell me, you can ask me, what is huge amount of data? What is the example of huge amount of data, which is also known as big data? So when your simple statistics is using huge amount of data, we will call it data science. As simple as that. Before, we will call it statistics when we are having a small sample size. Now, if we have a sample size like the parameters of the whole population, or the number of messages that is being sent in a this, what do you call, is social network. Okay, types of likes, number of likes that is being given for a particular kind of this thing, what do you call, what you video, or a particular kind of this thing, what do you call, image, a particular kind of music. Okay, so when we use a lot of data with the same principles of statistics, we will call them data science. Okay, for example, I do not know whether you are aware or not. Many a times, people will say, consider that there is a person who usually do YouTube for business. So he will be concerned about something called YouTube algorithm. So he should know which kind of videos are more likes, which kind of video gets more likes, which kind of music you should put. Understood. But he do not know about all these things, but he knows the terminology algorithm. Now, what is this? It is nothing but the data science that YouTube uses. YouTube try to figure out what is the length of the usual video that is liked most. Okay, when it finds out that, okay, if it is within 10 minutes, everybody watches it. So what YouTube will do is, if you uploaded a 1 hour video, this algorithm will put that thing in behind. But on the same topic, if somebody has made a 10-minute video, it will come forward when you are searching for the particular content. Coordinator madam, have you informed the rest of the participants to join immediately? Till now only 10. Coordinator madam, please one more time you share that the class is going on. Please join. Understood. So this is data science, means statistics with huge amount of data. So one more example is how many persons are coming to a particular mall and which kind of shopping they are making. Like Amazon also uses algorithm. Okay, so it finds out by inherent mechanism that which kind of products are being sold and which kind of products are not being sold. So the products that are not being sold will be this thing will be like what will be listed with a discount or some coupon or things like that. Okay, there is a service called AWS. How many of you have heard about AWS? Write in the chat box. कौन जानता है? Okay, Shayak Mondal has heard about AWS. Nobody else? Okay. So AWS is a cloud service which is provided by Amazon. So what exactly it is doing now? It is basically gathering a huge amount of data about all possible things that is going on the earth. Okay, all possible things. Not only this thing, it is getting track of streaming services, behavior of persons, um everything. How much people are getting sick, what is the age group in different countries, which people seek domestic help. Um, uh, then you can ask me, sir, how AWS is getting this kind of data, why everybody is sharing that data? Because when you search something in Amazon not only understand that you are searching it, it also understands that what is your domestic need. Okay, if you search for a like some kind of orthopedic equipment, the Amazon will find it out that in your family or in this particular area, there must have been some kind of trauma, and therefore you are using that orthopedic belt or something. Recently, I purchased one for my mother. So Amazon has not only recorded that I have ordered that, in its intelligence, it has been understood that there is somebody who requires an orthopedic belt. So next time when I'll open Amazon, no, it will propose me like this move gel or some magic spray or some heating coil or something related to my search. It is also getting data from this thing, what do you call as Alexa. So if you order Alexa that play this song, Alexa, for my kid, so Alexa will find it out that there is a kid in the house which likes Chuchu TV, and therefore it will start proposing kids stuff to me. So this is how the overall picture is. So as a biomedical scientist, our job is to harness this kind of technology for our benefit. Okay. So as I have given you one lab visit, no lab demo. It's some other lab where you can see a lot of investments in terms of what you know, this equipments, reagents, electricity, and many more. The same experiments no, we are doing. We have done all these 3 days. Similar kind of experiment in silico. Okay, so you cannot challenge the precision also. The precision is also very high. Why? Because many people have tested the algorithm, and over the time the algorithm has also increased its capacity by getting all the data. Okay, because I used albumin, nobody till now used albumin, so that albumin data is also inserted into its database. Okay, in albumin this kind of this DNA binding residues are there. Okay, so I think you can understand the the complexity of the algorithm and complexity of the system, as well as its ability. So randomly people are saying no, that artificial intelligence will do a lot of harm. Why? Because people are skeptical, because its ability is so huge that tomorrow it may happen that nobody requires a human intervention. But trust me, that will not take place in near future. When a complete human intervention will go, that is not possible. Yeah, that is possible for petty things, very very nominal things where you do not need any high level of intelligence. Let me give you one example. Robotics. In robotics, what happens? You know, you give pre-formed instructions, and one robot comes and does the thing. So if you keep it like that only, it will give you a feeling of a domestic help. Okay, until and unless you are putting a gun in front of him or telling that this is a gun and this does this, isn't it? So it's up to humankind that how exactly we this thing use this technology. And despite of it, there are some drawbacks, and I'm sure that over the time, no, we can able to fix these groups. Okay, now coordinator madam, one more time we'll do the same thing. Madam, select both the columns. Yeah, select both the columns. Now when we are selecting, we are giving some instruction that this is my training set, and on this basis I want to do the unknown calculation. Okay, there was unknown calculation, right? So what we'll do is we'll go to the insert, option, and then go to the this chart menu, the second chart, XY. Let me see if this is XY. This is XY. Click on it. Then go for the this one. Correct. Okay. Now what you do, you select the curve. Acha, no, before going to that, I do not know all of you are present or not. I cannot see some. So what exactly happened now? What does this thing indicates? This indicates that by and large there is a straight line equation. By and large, it is like fragmented. There are some seasonality. This is called seasonality. Okay, seasonality means due to some reason there were some exactly not straight line relation. That is by far a straight line only, but not exactly a straight line. Now, what is our job? Our job is to reduce the noise. Okay, noise is what? Noise is the outliers. Okay, so what we'll do is, I'm sure those who are in senior classes or who are PhDs, they must have used this kind of tool many a times. But in any data science class or any machine learning, this thing, no, people should start from this. So that their understanding will become very, very lucid. Okay, so we'll reduce the noise and reduce the seasonality to make it into a perfect straight line. So in order to do so, madam, I'll request you to click on the this thing tab so that all the points are selected. Fine. Then right click. Now what we'll do now, we'll try to figure out the equation of it. Then add a trend line, fourth option, third, fourth, fifth option. So you click on this. So when we click on this, there must have some equation. Acha, madam, you repeat this task again. You please select the curve so that all the points are selected. Then right click. Add the trend line. Oh, I see. Okay, okay. Now, madam, here no, you write. There is an option that we have already selected linear. Madam, on the left pane, format trend line. You scroll down a little, scroll down in the format trend line window. Then display equation on chart, the third option. Then do OK. Okay, fine. Now what you do now, madam? Okay, fine. We are having this equation now. Now this is the equation which will give us the ability to predict. Okay, now this is the ultimate goal of all of computer science. Ultimate goal of computer science is what? The ability to predict. Now here you see how easily we have acquired the ability to predict. Okay, now we'll predict. Now what we will predict? We will predict if the luteinizing hormone concentration in serum is 147 international unit per ml, what would happen? Madam, write it down, 147 international unit in LH. This is your number only. Fine. So now we have to, no problem. Now what we'll do is we have to do the calculation. So let us see here, there are two axes. Okay. So from 0 to 400, this axis is called x-axis, which indicates the luteinizing hormone concentration. And there is one more axis, which is from 0 to 100, which is called y-axis, which is nothing but the number of polyploids. Remember? Okay, remember. Now what we'll do is, we'll calculate what we should calculate, who can tell me, x or y? What we should calculate?
You can go with the Y.
Y. Okay, fine. So we'll calculate now. Yes, I know, Saif Kondal. Because what happened, I do not know whether you have this thing noted or not. One of the student was feeling like ahead of time, and she had given a feedback that I should go a little slow. So looking at her, we are once again repeating. She was not able to follow it. Seems. Okay, so I know we'll come back to that. Do not worry. Acha. So what we'll do, madam, you do it right here only. We'll find out why, and for that, what we'll do is 0. You open one more window and put a equal to sign. Madam, any window means any cell. You select any cell and in that you put a equal to sign. Any window, let it be, no problem. Equal to sign. Start with an equal to sign. Fine. Then you write 0.2293. 0.2293. There is no minus right now. 0.2293 have you written? And then put a star, means multiple sign. Then you write 147. And then put a enter. Okay. Now one more time, one more equal to sign. One more equal to sign. And then here you write 32.013. Here you write 32.013. Then give a plus sign, plus. Then select the other upper, this thing, upper cell. Then go upper cell. Click, 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 click. Then put an enter. Now, so what does it mean is, are you getting? So if you have a blood concentration, serum concentration of luteinizing hormone 147 per international unit per ml, then there is a possibility that you will have 65.7201 numbers of polyploids under a FNAC observation or a biopsy. In biopsy, what happens? We have already spoken. To identify the polyploids is the major task. Okay, some other tasks are also there, but for the sake of the simplicity, what it was? Just hold on. What was the message? Sir, it was 12.01. Which one was 12.013? No, it could not be 12.013. Why? No. I think some mistake. Just let me check. Anyway, you figure it out. You should calculate y. You should find out y. Okay. So for that, what you should do is you should do 147 into 0.2293 plus 32.013. Okay, whatever comes. Okay, so this is how the prediction is done. So in order to do this prediction, we have to have the foundation of statistics on which data science is being built, where you are having a lot of data. As you know, many a times you would have seen that we are using a lot of data. Okay, now this data is called the training set. On the basis of the training set, your prediction will be formed. Okay, now why machine learning? Why we have not stake to the data science on? Why machine learning has developed? Machine learning has developed because in data science there are some applications in machine learning which uses multiple types of statistics altogether. Okay, multiple types of, this is a simple regression, no. So there may be some mistake in real life. You will see that it is less or more or something, not exactly this, isn't it? Why? Because we have used the linear regression. So there are 4 different other types of regressions also, so we have not counted that. So who will decide that which kind of regression model you should follow? There should be some directions, no. So why I have used a linear regression model? Because it is the most easy to understand. Otherwise I have to do logarithms, multiple other computer and mathematics, so I'm avoiding it. Okay, so this is the reason why you are not only sticking to data science, but you have moved a little bit higher in machine learning, where you are integrating all the data science and making one more package which uses a huge amount of data and giving you very near precise predictions. Okay, let me see the, if you want to say something. Okay, Aditi Mehta, please once again you understand this, that this is the equation. Okay, y is equal to 0.2293x plus 12.01. Okay, so in the meantime, I do not know whether you people have some requirement or not. I am taking some project fellows, remote project fellows. Okay, those who are interested, they can contact the coordinator and all, because I do not have your direct contact. Project fellows means it's a this I'll not able to provide you any fellowship if anybody is interested. But what we can do is we can eventually do a lot of research together and eventually do some intellectual property as okay, where I'll be giving you a specific task and you have to complete that task. And what is the this thing is your this thing will be getting published in the very good journals, the way we publish usually. If it is okay, so if it is for you any use, then you can definitely contact this company. Achha, now coming back to this, so now in machine learning, no, in machine learning and human learning is also equal. So I had given that example also. If there is a flame, you are putting your finger will burn. Like how the kids will understand this, because they have not experienced. So once they'll experience, they'll get it done. Now that is called unsupervised machine learning. Unsupervised means nobody will tell you whatever. In a room, you left the child, and the child is this thing growing all by itself. You are doing nothing. You have only given the resources. Okay, you have given a process through which every time food comes, you have given something so that dress comes. If the child understands that okay, I need to eat, the child will eat. Understood? Unsupervised algorithm. So you can ask me, sir, which one is good, supervised or unsupervised? And supervised, what happens? You have some basic idea that what is the kind of result you want. Okay, for example, I want a robot who will only cook for me and clean all my utensils and my clothes, but I don't want him to dress me. I don't want him to serve me my food. I'll take my food and I'll eat. I'll dress my dress. He'll clean it. The robot will clean it, but I don't want him to like this thing. In those cases, no, supervised algorithms are great. Okay. Now you can ask me, sir, where I should use unsupervised algorithm. Now, unsupervised algorithm should be used where till now nobody has any idea that what will be the outcome. Okay, let me tell you example. Chess. You are starting a chess match. Okay, so nobody knows what. Nobody can tell you after one move if you give a particular like Sicilian defense. You're doing Sicilian defense, and nobody can tell you with precision that if you start with Sicilian defense, you'll win the game. Okay, if somebody starts with an Italian, what would happen? Nobody knows. From first move, nobody can tell you that okay, after 30 moves, the match will be over. Are you getting? So this is the thing. Now coming back to that, okay, in unsupervised algorithm is something like RAPDPC. Okay, who can tell me what is RAPDPC? As I have said, we will extend this session a little, and in the end, we will do the tools and we will also see the troubleshooting. Who can tell me what is RAPDPC? Why I am giving this example of RAPDPC? So in RAPD PCR means you have identified an animal or a plant which nobody has seen till now, and it does not resemble anything also. Okay, but you want to know which kind of species is this. At least you want to this thing increase the number of the DNA, because very small amount of DNA you isolate. Okay, consider that you are a this thing deep sea diver, and eventually you found one this thing organism which does not look like a fish, but it's a live organism. It also does not look like a this thing. What else? Plant. So what you will do? But you want to this thing multiply the DNA because you want to preserve the germplasm, and you also do a little this thing what you call is barcoding of the animal and try to figure out which kind of protein it produces. So in that case, how you will multiply the DNA because you do not have any primer. As you know, no, you can do PCR only and only if you have a primer. If you do not have a primer, what is the this thing of a primer? The primer should have sequence homology to the 3' ends of the target DNA. Now, what would happen if you do not know the structure of the this genotype of the animal? What would happen? You have no idea of the 3' ends of the target DNA, isn't it? So you cannot able to PCR. Then at that point of time, what you do? You do random primary. Okay, whatever kind of primary you have in your lab, you put everything. Some or the other way, it will stick somewhere. If some sequence homology is there, and eventually it will start doing the multiplication or polymerization, whatever you say. So it is something like that. Okay, so in those cases, you should do unsupervised algorithm, which we generally do not do. But in bioinformatics, we do not do because it is of no use. We must have to have some rational this thing prediction. So most of the time, you will see that we are using supervised algorithms. This example is support vector machine, artificial neural network, mostly artificial neural network, support vector machine. Tomorrow will teach you mm, uh, you make notes. This hidden Markov model. Okay, these are also supervised algorithms. So tomorrow we'll use a tool where you will learn how hidden Markov modeling is usually done for bioinformatics, protein structure analysis. Okay, so what happens? This is how this thing happens. So, madam, now you can close it and then share the PPT one more time. One more time, we will give them some examples and then the very important snippet that you shared previously. How exactly these algorithms are being built. One snippet is there, nice snippet. Everybody should take a snapshot. Next slide, ma'am. Now, this is a very good schematic representation, we shall say, of a software. That how exactly this all. Now, this is a basic or primary architecture of any software, bioinformatics, for biomedical applications. This is the framework. Okay, then you change many things, but the framework remains same. So let us see. In this tool, we used to do interleukin 13 prediction. Okay, if any peptide can stimulate interleukin 13. So as I have said earlier, interleukin 13 is a pleiotropic interleukin. It can do multiple different things and very important for immune response. So what we will do, we will use a training set, nicely given. If you see this, no student is there who cannot understand machine learning for biomedical research and technology. So here nicely it's been given that you should have a training set, positive training set, negative training set. Now, as you can see, positive training set are little less. So the positive training set is less. So if you want it more robust, robust means what you know, there is no false negatives. False negative means this is not an interleukin 13 producing peptide, but despite of it, it is showing positive. So if you want to reduce that task, sure. Mary, you can screenshot and you can definitely take it from the coordinator. No problem. So what exactly happened? This is this thing. The training set could be increased. Now here you see negative training set is increased hugely. Now this is also working hand in hand. So if you know something is negative, no, that is also a positive for you. So that is what I say all my students. So if you get a negative result, try to publish that way. That also. Why negative result is also a result only. Okay, you if you find that, you just have to write in a way that the reviewer is convinced. Okay, negative result, you cannot, no sir, this particular this thing is not able to destroy the bacteria, so no use. No, you add either you report it individually or some other place where it is relevant, this negative result. So it is not that negative result is just for getting rid of it. So here you see, in negative data set there is a huge data set. This is also positive. Okay, why positive? Because if you know that this particular sequence is not working, that is also have some use, isn't it? The algorithm becomes more what you call powerful. So do not worry. But if you want to increase the this thing what you call specificity, you have to increase the positives. Okay, but the efficiency have no this thing change. It is not that if you have positive data set more, then efficiency will increase. No, if the threshold, there is a threshold, efficiency means what? In 1 second, something like turnover number. In every second, how many results you can find out? How much time your algorithm is taking to find out the result? Now that is called turnover number. Now that turnover number is nothing but the efficiency of the algorithm. Okay, can you remember one algorithm was there? It was getting stuck. It was not able to show the result, and therefore I put my email ID. Despite of it, it did not respond. Means what? Means its efficiency is very low. Many a times this thing also happens. Anyway, so coming back to this, and then what we'll do? We'll do the pattern recognition. Pattern recognition means what? Feature extraction. So there should be some similarity somewhere. As I have said, pattern. That pattern is understood, and that pattern has been made into a separate this thing, separate what? Separate processor. In one processor, all the data. In another processor, only the features. Okay, I want this pattern. Okay, it is something like your which kind of dress you like. Somebody likes a tight jeans, so whenever it sees a tight jeans, it will take it. But somebody likes it this thing comfortable fit. So if you see a comfortable fit, you will take it. Are you getting? This is called a pattern recognition. So next is what? Next is feature selection using MRM. It's a particular inbuilt mechanism. And next you see the names of the algorithms that this thing has used. Random forest, decision tree, support vector machine, then some more, then KNN and XGB. These are all different algorithms, not very popular. Anyway, decision tree, random forest, support vector machine, these are very regular kinds of what do you call this supervised algorithm. Then what we do? Then we do best generalized model, as I have said, algorithm. Now this is the algorithm. Now this algorithm, what it will do? Now it will give the testing data set. Okay, some test it will do. Now for test also you will give some positive, some negative. Then you will see whether your algorithm can able to correctly predict it or not. So when you see that the prediction ability is more than 80%, 90%, you published your result or you do a patent or do whatever. Are you getting? Now, if your idea is not very new, then you will not able to patent. Interleukin 13 prediction, similar tools are available. So this fellow has not submitted any patent. So just simply this thing, what you call this thing, put it into a paper. Understood? Going to the next one, madam. Next slide. That was one more example of this thing, artificial neural network. Okay, I am sharing one more figure to our madam, this coordinator madam. The coordinator madam will give you a snippet. No, you will be amazed to see that how simple the algorithm is. The concept is so simple. The application is very tough. To build a artificial neural network is very tough. But it's not our cup of tea. It's for computer science people. But the concept is so lucid that any student, be it a class 5 student, can also understand what is artificial neural network. So I'll share this thing to the coordinator. One snippet is there. Just hold on. Madam, please share the snippet that I have shared with you. You can read it, you can this thing this. Uh, Share the snippet and then we'll go for the tools. Okay. The first of all we'll do the troubleshooting and then we'll do today's tools. Okay, so I could not this thing it. If you see the snippet, no, you will come to know that how how all these algorithms works. Now this is this thing custom for artificial neural network. But by looking at it, you will get all pictures clear. That what exactly is machine learning, on which basis this is working on. Okay, and then we'll go to the troubleshooting of this thing protein binding these things, oh sorry, DNA binding proteins, and then we'll go to the today's the other one. Okay, here if you see, no, you increase the size. Okay, if you increase, no, can you able to see what is the training set? Fine. Now this one on the middle picture, no, leave those two, these are some applications. I'll put the middle one. So participants, if you see here how these algorithms work, okay, we are not giving him the exact 8 because we do not know the 8. We are about to predict what we give in training set, something similar to it. Are you getting? And then there are multiple layers. Layer means what, you know, as I have said yesterday. So this is only a similar types of figures many more times. Okay, with combination of something. From that combination also, it should identify what exactly is given as training. Finally, it will able to identify the digit 8. Okay, so if you train your algorithm with similar results, then what it could identify? It could identify the pattern. Okay, now this initially what you have given. Okay, digit 8 should have this pattern. Not exactly the same, but similar pattern. Whenever you find this kind of pattern, you say that this is digit 8. Okay, so once the algorithm has been trained with these data, what it can do is it can eventually find it out what is digit 8. But many a times, as I have said earlier, that it can get confused with digit 8, digit 6, and digit 0. Sometimes, not every time, many a times, isn't it? So those problems are there, but once we increase the training set some or the other way, the algorithm becomes more robust. So this is what I wanted to share with you. Now we'll go to the troubleshooting. My audio was there. Give me one more this thing. If audio was there, right?
Yes, sir.
Okay. So now we'll do the troubleshooting. Coordinator madam, the links that we have provided, this autoclav, please share this. Please share the link by clicking on it for autoclav. Okay, now this was the study regarding antibiotic resistance. Okay, I'll use this study for our new tool. Madam, I'm talking about the old tool where you can have the autoclav this thing sequence I have given to you. In your WhatsApp, it is there. No worry, we will come back to this later on.
Okay, sir.
No, why not audible? I'm quite audible, I guess. I put a lot of energy in my sessions. So I hope it will be definitely be audible. If something is not wrong from your end. Uh, Otherwise, what you do, Shayak Mondal, if you have the link, no, share in the chat box so that it becomes easier for the madam. share it in the chat box, that link, and then I'll go ahead so that madam can click the link from there itself. Can anybody share the link? Great. Srijani Shen has shared the link, madam. Please click on it and then go to your yes. Fine. We'll see why this thing is there. Then we'll change a little bit. Fine. So in this one, madam, we'll go to service. We'll go to service. We will not download a local copy. Now here what we'll do is, now predict license. Okay, madam. Now go to one more new window where you write NCBI protein. Where you write in Google NCBI protein. In one more window. Fine, absolutely great. Okay, now in this window, madam, you write it down. DNA polymerase. Then put an enter. Search the lowest size of protein. Do a little this thing. See, if anything less than 800, nothing. Go. Down.
The third one, 575.
Okay, go to FASTA. Yes. Click on FASTA. Now, FASTA is nothing but the simplest format of amino acid sequence or DNA sequence. So as you all know, a protein sequence is nothing but the linear sequence of amino acids that are connected by a peptide bond. I did not wanted to discuss all this because I thought that at least many people are PhDs and all, they may feel this thing. So I did not discuss. But I got a feedback that I must have discussed all this. So what is FASTA? FASTA is nothing but a simplest possible text-based format of amino acid and DNA sequence. Now, from where the DNA sequence comes? From a DNA sequencer, or from PCR also you can do. And then there are multiple different levels. Today we are having NGS also, where by means of a shotgun shotgun sequencing, you can do a sequencing of a huge amount of DNA, because each and every nucleotide is being treated with a particular kind of fluorescent tag. Every particular this thing, whenever a nozzle come across with an adenosine, it gives a particular kind of peak. Whenever it comes across a guanosine, it gives an entirely different kind of peak. By looking at the peaks, you can identify where which nucleotide is there, and by doing so, huge amount of sequencing can be done very easily. But however, the funda remains same. Senger's method. Okay, by dideoxynucleotide, we are using the same thing. Okay, the funda remains the same, but the process has been changed. Similarly, for protein sequencing also, we will do Sanger sequencing only, because Sanger was the only one person among biomedical scientists, not physics, chemistry, and all else, who have received a Nobel Prize twice. First for DNA sequencing, second for protein sequencing. Coming back to the point, we will select at least 3 lines, madam. 3 lines at least we select. Okay, three. Copy. Then go to the tool. Paste. In the first M, no, the first M is there, no, madam. Before that M, you put a greater than sign. No, less than sign. Okay, then run. Then go to run. Let us see what happens. Okay, it will come back. Don't worry. It will come. Length 150. Position 26R. Position residue 26R. Invalid sequence. Invalid sequence. Acha. Why this is invalid sequence? Why? Hmm. Acha, madam, go to services again. Go to services again. Then load example. Click example is there. Example, example. यहाँ पे नहीं, ऊपर ऊपर चले जाइए. हाँ, क्लिक कीजिए. Just 1 second. अच्छा, oh, Bob. Okay, we can only put a single line. Madam, delete that. Reset. Reset button दबाइए. Reset button दबाइए. हाँ, go to that this thing sequence. Protein sequence, madam. हाँ. So end terminal end. End terminal end वाला लाइन. पहला वाला लाइन. हाँ, कॉपी कीजिए. कॉपी कीजिए. पेस्ट. लेस देन साइन दे दीजिए. लास्ट के जो 3 है उनको भी डिलीट कर दीजिए मैडम. यहाँ पे जो 3 है. चलिए अभी रन कीजिए तो देखते हैं. Okay, now how it has made it, I think everybody is clear on which basis this algorithm can find out. अच्छा. Madam, would you please once again go to the services? One more time example. अच्छा, अच्छा. Okay. Madam, reset again. Reset. Then you write here test one, TEST. Greater than sign? No, greater than sign, madam. Oh, sorry, less than sign. Test one. Now come, put a enter. Now you paste. Now F and I you delete. Now run. So I'll come back to you all with this. But that day, this was not the scenario. Last time, what we did, what I used, I forgot. Achha, anyway, what I tell you is, what you do know, you select the whole starting of the FASTA and then only single line, no problem. In end terminal residue, if it is there, then it will definitely find it out. And the whole length is may not be many a times necessary. But if you want to do throughout this thing, then this particular tool will give you limited results. Okay, there are more robust tools, but this was only for to make you understand that how these algorithms work and how by using a web server you can replicate your this thing experiment in a very short period of time. Now coming back to today's this thing, madam, now you share the paper that was on this thing, biofilms. You just click on it. I can see it, antibiotic resistance. Here you click on antibiotic resistance. So here in this case, what we have done is we have tried to figure out that in E. coli, that means coliform group of E. coli, how biofilm production is enhanced due to its architecture of the plasma cell wall and cell membrane. Okay, so we got it published recently only. थोड़ा सा नीचे जाइए मैडम. वो तो देख लिया। थोड़ा नीचे जाइए, नीचे जाइए, इस साइड में आइए, थोड़ा फिगर फिगर जो है उनको दिखाइए। कैन यू सी? उसके थोड़ा, हाँ, these are different figures that are already there. There is a wet lab, there is a dry lab, and then there is a combo, and then there is the obligation. Okay, so this is the ideal situation where you have a little wet lab data, and you are validating it with a dry lab data, that means by simulation, and then eventually your result is much more validated. Understood? Now it also got cited also, no problem. This year only it got published. Now, madam, we will do today's task. I have shared this thing, I think. Oh, I'll share the link. I'll share the link. No problem. Just 1 second. I'll share the link, and very quickly we'll able to do it. So today's this thing, the tool is for a completely different kind of this thing. What do you call is method and application. Okay. Now this is the tool. I'm share it in the chat box itself. Okay. Yes. Can you find it? Okay. So today's tool is based on support vector machine. Support vector machine, as the name suggests. With some support, it will do a vectorization. Vectorization means, you know, prediction. Means today sales is this. Tomorrow what will be the sales? Today growth is this. Tomorrow what will be the growth? This kind of vectorization, extrapolation in clear terms. So today we'll use this tool, which is based on support vector machine, and it will try to figure out what is IC50 value of a particular drug. Okay, now again, what is IC50 value? IC50 value is the value which will help you to understand how to kill some bacteria at a half concentration. Means what? For example, if if the IC50 value of any compound is very low, it means that with the double of its concentration, all the bacteria present in a particular area will be dead. Are you getting? The smaller the IC50 value, higher is its potency. Okay. Now here you remember one thing. There, as I have said yesterday, I do not know. Please listen carefully. There is a what we call is evolutionary mechanism through which bacteria becomes resistant to drugs. Okay. So one of the mechanism is biofilm formation. Biofilm is nothing but making a barrier, physical barrier, not molecular, physical barrier, exterior to it. So the antibiotic cannot able to penetrate inside. So this particular tool, listen very carefully, you can use it randomly. Whatever kind of data, if you have some microbiology lab or something, no, you can use this tool like anything. It is based on support vector machine. So what we'll do is we will identify a molecule and see what is the possible antibiofilm activity of that molecule. Now, madam, I'll request you to one more time go to Google. Go to Google. Ah, madam, go to yeah. Here you write drug bank. Again, yesterday also, and last time also we did drug bank. Ah. Now click on it. Okay, where is the search window? Search. As you can see, here you write. Here you write crystal violet. Crystal violet. Crystal violet. As you all know, crystal violet is a stain, but at the same time, it has proven its antimicrobial efficacy. Okay. Anyway, why I have taken crystal violet? Because we are doing a similar kind of simulation in the lab. To study antibiofilm activity, crystal violet is also used. So everybody throughout the world is using crystal violet to identify whether a particular bacteria is able to produce biofilms or not. Okay, so we are proposing what? We are proposing that crystal violets, due to its inherent antimicrobial activity, it may destroy some of the microbial architecture of the biofilm. So it must not be used anymore. Anyway, that's a long-term goal. We have to prove it with foolproof this thing. Okay, so what we'll do is we'll go to download. Madam, download पे चले जाइए. Can you see the download button? Down नीचे 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 structure का नीचे. हाँ 3D download. हाँ click. Then SDF will come. SDF one SDF. हाँ click. Okay. Now copy it. Control A. You put Control A. So everything will be copied. Then copy. Control C. Go to the tool now. Then paste. Control V. What is it? All these things कहाँ से आया? चलिए ठीक है देखते हैं. सबमिट कीजिए तो मैडम. Participants, you also do. Let me know if you get some other result. Oh, okay. We got it. So inhibition efficiency is not very high. Inhibition efficiency IC50 value by means of SBM is 0.05. Okay, that's why everybody is using. It's not very dangerous. Okay. So this tool has predicted that this particular thing, what is crystal violet, is not very destructive. Okay. So we'll look for it that with some other tool, what exactly it says, what are the other literature is saying. Are you getting? So this is how you should plan your research work and all. So as I have already spoken to you, if any of you want to do a little bit more with the internship and all, you can definitely get back to us. Either by this thing, you can contact the organizers also. However, I am also giving my email ID and my WhatsApp as well. Tomorrow we will talk more about vaccine design, reverse vaccinology, antibody design, mainly immunoinformatics. Okay. By saying so, I'll call it a day. If any doubt, you can ask me.
Hello.
Okay. If there is any doubt.
In case I need any clarification, can I WhatsApp you, please?
Why not? Definitely. Put a +91 if you are from outside India. You should
Yes.
put a +91. Sure, you can WhatsApp me.
+91.
Then 84140
Uh-huh.
39472.
Okay, thank you. Definitely I will.
Okay.
Thank you.
Yes. So if any more this thing is not there, we'll call it a day. Okay. Thank you.
Thank you, sir.
Yes, thank you.